Hello all. So let's see the part 2 video for animal kingdom. So before going into the animal kingdom, we so far have seen that what are the phylums that been divided the animal kingdom. So entirely we have 11 phylum. So once again I'll tell you the 11 phylum and how this 11 phylum got separated. So phylum Porifera, phylum Cnidaria, phylum Ctenophora, phylum Platyhelminthes, phylum Asclehelminthes, phylum Annelida, phylum Arthropoda, phylum Mollusca, phylum Echinodermata, phylum Hemichordata and phylum Chordata. So let's see the basis of classification. So how the animals are being divided in this category. So the basis for classification we have to remember level of organization and circulation, and next coelom, symmetry, so germ layers, so as well as the notochord, cephalization. So these are all the things we have to consider, and we have to divide the animals into 11 phylum. The very first thing we are going to discuss is level of organization. Obviously, if you are talking about level of organization, we will remember about cell. So the group of cell together we call it as tissue, and the group of tissues together we call it as organ. The group of organs we call it as organ system, and all the organ systems together will form the organism. So this we call it as the level of organization. So on what level the organisms are there, based on that they got divided the phylum. So the very first phylum, that is phylum Porifera. So that is the only phylum will have cellular level of organization. Okay, that is the only phylum, because it will be like an aggregate of cells. So like we have conocytes and we have the archaeocytes. We have many different types of cells in Porifera, that means in sponges, doing many works. So that's how it will comes under cellular level of organization. And Cnidarians and Ctenophora will comes under tissue level of organization. And next, Platyhelminthes and Asclehelminthes will comes under organ level of organization. And from Annelida to Chordata will be coming under organ system level of organization. Okay, so this is one of the basis for classification, that is level of organization. Okay, and remember only one kingdom that comes under cellular level, that is Porifera, none other than the sponges. And next basis of classification, that is circulation. We have two types of circulatory systems. One is open circulatory system and other one is closed circulatory system. As the name suggests, the open circulatory system. So in the open circulatory system, the blood will not be pumped into blood vessels, and there is no blood vessels in the body. The blood will be flowing directly into the sinuses, are the spaces present between the body wall and the organs. For example, if you accidentally have seen cockroach, stepped on the cockroach, suddenly you can notice a white color fluid that is coming out of the cockroach. That is none other than the blood. So usually insects will be having open circulatory system. I hope you got the point. Insects will be having open circulatory system. And next, because there is no blood vessels for insects, so for Arthropoda. So even for mollusca also it is open circulatory system. And next, closed circulatory system. As the name suggests, the blood will be flowing directly in the blood vessels. So that won't be any spaces like that. That will be very much organized. For example, we humans, we have closed circulatory system. So like birds, reptiles, aves, all these things also will be having closed circulatory system. The blood will be flowing in the blood vessels. Okay. So I hope you got this point. This is the second basis of classification. Next, the third basis of classification, that is segmentation. Okay, so the true segmentation or metameric segmentation we call it as, or we have one more category, that is pseudo segmentation. What is the meaning of segmentation is, you will be having externally and internally separation of the body parts. So that too equal separation has to be there. That we call it as segmentation. You can find the true segmentation in Annelida because the body parts got divided, because the organism got segmented externally as well as internally. But you can see only external segmentation in Arthropoda. Internal segmentation is seen in mammals. These are the points you have to remember regarding segmentation. And next, very important thing, so the fourth one, that is symmetry. Symmetry is nothing but arrangement of the organs inside as well as outside the organism. That is based symmetry, will find out that we have three very important varieties of symmetry. So one is asymmetrical. Asymmetrical means if any plane passing through the center of the organism, you cannot make the organism into two equal halves. That we call it as asymmetrical. So example for asymmetrical are sponges, snail. Snail also will comes under asymmetrical. Sponges also will be asymmetrical. And next, so we have like corals are asymmetrical. So there are certain organisms which you can't divide into two equal halves. And next, radial symmetry. Assume a cake, round cake, which you can divide into equal parts by any plane passing through the organism. That we call it as radial symmetry. Example for radial symmetry is jellyfish, octopus, Tinoplana. So these are the examples for radial symmetry. And starfish will be exhibiting pentamerous symmetry because it has the 5 limbs. If you cut through the limbs only, you will be getting the equal parts. And next, so bilateral symmetry. Bilateral symmetry means, for example, if you take humans, so there is only one plane. If you cut in that plane only, you will be getting an equal halves. So that too in the center sagittal plane. If you cut the organism in the sagittal plane, then only you will be getting an equal parts. So that we call it as the bilateral symmetry. Lateral means sides. So you will be getting two equal sides. So that's why we call it as bilateral symmetry or biradial symmetry. So that is about symmetry. And next we will go with coelom. So what is coelom? Before going into the coelom, we'll go with the germ layers. Usually at the time of gastrula or gastrulation, the entire organism, so when from the zygote you will be getting this gastrulation. So there will be 3 germ layers usually for us. So but we have called ectoderm, endoderm as well as mesoderm. Okay. So ectoderm is the outer germ layer, endoderm is the innermost germ layer, and mesoderm is the middle germ layer. But for certain organisms we have only 2 germ layers. So those organisms we call it as diploblastic organisms. There are certain organisms which have 3 germ layers, that we call it as triploblastic organisms. So coming to the diploblastic organisms, for example, so under having only ectoderm and endoderm. So diploblastic means having ectoderm and endoderm. So that is ectoderm and endoderm is for sponges and cnidarians, Ctenophora, Platyhelminthes. These are the first 3 are diploblastic, Porifera, cnidarian and Ctenophora. So are having diploblastic nature. And triploblastic nature will start from Platyhelminthes. Platyhelminthes to till chordata, you will be having triploblastic nature. So ectoderm, endoderm and mesoderm. First 3 phylums are diploblastic, and from the topic of Platyhelminthes till chordata will be triploblastic nature. Okay. And next we'll go with the coelom. So coming to the coelom. Coelom is nothing but the cavity that is present between the body wall, the inner body wall, and that is the cavity that is being formed in the mesoderm, which is lined by the mesoderm. That we call it as the coelom. So actually the organisms such as Porifera is acoelomates. Porifera, cnidaria, Ctenophora and Asclehelminthes. First 4 phylum will comes under acoelomates, will not have any coelom. and pseudocoelom will be there for only one phylum, that is Asclehelminthes. You will be having the coelom as pouches, which is lined by mesoderm, but it will be having like pouches, which is not a true coelom. Only Asclehelminthes is the phylum will be having the pseudocoelom. And next, from Annelida, the true coelom will start. From Annelida to Chordata, the true coelom will be seen. So those we call it as acoelomates. And next, we'll go with notochord. So what is notochord? Notochord is nothing but a dorsal hollow tube which is mesodermally origin. So if the notochord is present, we call it as chordates. If the notochord is absent, then we call it as non-chordates. So it is very simple. From porifera to echinodermata will comes under non-chordates. And chordates are otherwise consists of notochord. Okay, so this is based on the presence and absence of notochord. One more time, so basis of classification will be level of organization, circulation, and we have the symmetry, germ layers, coelom, and we have the segmentation as well as notochord. So that's it for the day. So thank you very much. I hope you understood. If you have any doubts, please don't forget to put in the comment box. Thank you, and please do subscribe my channel for more biology videos. Thank you.